do you feel about what President Trump said? John, I love you so much.
Who was it?
So sex is when a mommy and a dad.
Surrounding cytoplasms, stock
You take the A Lego, which is white, and match it with
In mitosis, the nucleus and its contents, most importantly, the two duplicated chromosomes, divide and are evenly distributed, forming two daughter nuclei. During cytokinesis, which usually begins before mitosis ends, the cytoplasm is divided into two. The combination of the mitosis and cytokinesis cycles produce two genetically identical daughter cells, each with a single nucleus, surrounding cytoplasms stocked with organelles and a plasma membrane. Each newly produced daughter cell may then proceed through G1 phase and repeat the cycle. This is usually through asexually reproducing insects and in healing of the human cells and growing of the human cells. Any questions?
No. How does it heal a gut and how does it heal the liver?
Take a look.
Mitosis is like a copying machine that makes identical daughter cells to the parent cell. So here you can see one William that is then copied into two Williams. That'll then be copied into a bunch more Williams by the process called mitosis that makes genetically identical daughter cells to the parent cell.
Owie. Owie.
In order for John and William cells to heal, they go through the process of mitosis. The first stage is prophase, in which the DNA condenses, organizes, and the shape of the chromosome begins to appear. The next stage is prometaphase. In this stage, the microtubules attach to each of the chromosomes. The next step is metaphase, in which the chromosomes align at the metaphase plate. Metaphase is followed by anaphase, where the chromosomes begin to separate. Finally, during telophase, nuclear membranes reappear around the two sets of chromosomes. Mitosis is now complete. After mitosis, two new cells are formed by a process called cytokinesis. As you can see, the cells went through mitosis and new cells were formed which allowed William's arm to heal.
Meiosis is a type of cell division that produces haploid gametes in diploid organisms. Two haploid gametes may then combine via fertilization to restore the diploid state in the zygote. Fertilization in meiosis alternate in sex life cycles. So sex is when a mommy and a daddy love each other very much. The daddy puts his sperm cell in mommy, and the sperm cell swims to the egg cell, creating the zygote. You get this, John?
No, I need a visual representation.
We'll show you a visual representation when we get back.
The sperm and egg cell made a zygote, and prior to the zygote being made, you could predict what traits the zygote would have using the gametes of each parent and using a Punnett square to map it out.
Yeah.
The Punnett square is a visual representation of predicting the alleles for the offspring. First, you take the parents' alleles and split them up into gametes. Next, place the alleles down their column or across the row. The resulting combinations are possible genotypes of the offspring. In this case, the capital B represents the dominant allele and the little b represents the recessive allele. You get a 3-to-1 ratio showing that 3 of the offspring will be dominant, 1 of the offspring will be recessive.
Another way to predict the genetic traits of offspring is by using a dihybrid cross. A dihybrid cross is found by taking the two crosses of the parents and finding their gamete combinations. To find the combination of each gamete, you take one allele and then match it with the other allele, the opposite one. Then you take the 4x4 square, listing each gametes on the top of the square and down the sides of each squares. You multiply them out in the same way as the monohybrid cross. Once you multiply them each out, you get each of your genotypic outcomes and each of your phenotypic outcomes. In this case, the genotypic outcome is just 4 of each of the alleles matched together, and the phenotypic outcome is based on which alleles are dominant and which ones are recessive. A sex-linked gene is a gene that is located on either the X chromosome or the Y chromosome. These are inherited based on the gender of the offspring. Since there are more X chromosomes, most genetic diseases are passed down from the X chromosome. In the case of colorblindness, it is on the X chromosome.
Who was it?
John, how do you feel about what President Trump said? John, I love you. John, I love you so much. I love you, John. I love you. You're the most beautiful human being I've ever seen.
DNA is made up of nucleotides. Each nucleotide consists of a phosphate group, deoxyribose, and a nitrogenous base. There are 4 different nitrogenous bases: thymine, adenine, cytosine, and guanine. As according to Chargaff's laws, these bases pair together, with thymine going with adenine and cytosine going with guanine. These base pairings come together to form double-stranded DNA molecules. In this double-stranded form, the DNA is useless. It must separate with the help of enzymes to form either DNA replication or RNA transcription. Both processes are essentially the same, with complementary nucleotides attaching to the ones on the DNA strand. The main difference is that in RNA transcription, is RNA nucleotides that attach to the DNA strand. RNA nucleotides are like DNA nucleotides, except that they have ribose in place of deoxyribose, and thymine is replaced by uracil. Single-stranded RNA molecule leaves the nucleus as mRNA. It goes to the ribosome, where every 3 nucleotides are read off as a codon. There are anticodons of tRNA attached to it and leave behind amino acids. These amino acids are chained together as more and more mRNA is read off, and eventually they form proteins, which are used throughout the cell.
A strand of hair was found at a crime scene. To figure out who the suspect is, we can use gel electrophoresis. You place the strand in the test tube, and then that is placed into a PCR machine, which stands for polymerase chain reaction. It's basically a technique that copies down the molecules of the samples. It is then heated and separates the DNA strands. The sample is then cooled in order for the primers to attach to each other. The process is then repeated until there is a usable amount of DNA that has been created. Poles are created in the gel that serve as a place to hold the DNA solution. Mixtures of different sizes of DNA fragments are then loaded into a well in the gel. The force that is needed to draw the DNA through the gel is provided by electricity. A power supply takes the standard alternating current electricity available from the outlet and converts it into the one-way direct current needed to set up an electrical field across the gel. After these samples are created, you can then compare them to the original model. Whichever DNA is closest matched to the first original strand, you know that it was a match to the original piece of DNA that was taken.